Question number 141. Match column 1 and column 2 and choose the correct option. In column 1, the very first option given is agarose. And as agarose is extracted from seaweeds, so A in column 1 goes with first option in column 2. The next option in column 1 is illusion. And as illusion is extraction of DNA from gel piece, so B in column 1 goes with fifth option in column 2. The next option in column 1 is ethidium bromide. Ethidium bromide is a staining dye for DNA, and so C option goes with fourth option in column 2. The next option is spooling, and spooling is when precipitated DNA is wound around a glass rod. So D in column 1 goes with third option in column 2. So the correct answer to this question is option number 4, A with 1, B with 5, C with 4 and D with 3. The correct answer is option number 4. Let's move on to the next question. Question number 142. Bacteriophages have very high copy numbers of their genome within the bacterial cells because. This is a direct line from your NCERT book, and the correct answer to this question is option number 2. Because their number per cell is high. This is the correct answer to this question. Let's read the rest of the options. The very first option say because their replication is linked to replication of chromosomal DNA is incorrect. The next option is because of absence of their own ORI sequence. This is incorrect. They have their own ORI sequence. And the last option, because of lack of vital genes, is also an incorrect option. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 143. PUC8 offers an advantage over PBR322, which is. So you have to find out an advantage among the given options. The very first option says presence of ampicillin resistance gene. As this gene is present in both the plasmids, this cannot be a correct answer. The next option is identification of recombinants requires simultaneous plating on two plates having different antibiotics. And this is an incorrect option because identification of recombinant requires single step of plating, that is plating onto agar medium containing ampicillin and X-gal in case of PUC8. The next option given is ability to distinguish between non-recombinant and recombinants based on production of color in presence of chromogenic substrate is a correct answer to our question because this is an advantage offered by PUC8 over PBR322. PUC8 recombinants produce white colonies due to insertional inactivation of beta-galactosidase, whereas non-recombinants produce blue-colored colonies. So the correct answer to this question is option number 3. The last option is larger size of this plasmid. This is incorrect because PUC8 is 2.7 kb in size while PBR322 is 4.3 kb in size. So the correct answer to this question is option number 3. Let's move on to the next question. Question number 144. If an alien piece of DNA is inserted at the recognition sequence of PVU2 in the E. coli cloning vector PBR322, the recombinant, and PBR322, PVU2 region is present in ROP gene, and there you have site for PVU2. If you digest this plasmid with this enzyme, this will lead to the inactivation of this gene, ROP gene, and based on this, the recombinants will appear blue in color. This is incorrect. The next option says become ampicillin sensitive but resistant to tetracycline. This is incorrect because ampicillin resistance gene and tetracycline resistance gene remains unaffected. The next option is will be ampicillin resistant but tetracycline sensitive. This is also incorrect because both ampicillin and tetracycline resistance genes remains intact. And the last option is will not replicate within the E. coli host. This is a correct answer to our question because rope codes for the proteins involved in the replication of the plasmid. So when alien DNA is inserted into the recognition sequence of PVU2, this causes insertional inactivation of rope, and therefore there will be unavailability of proteins for replication of plasmid. So the correct answer to this question is option number 4. Let's move on to the next question. Question number 145. Which of the following has been disarmed and modified into a cloning vector to deliver desirable genes in animal cells? So the keyword is animal cells. And the options given are PBR322, bacteriophage, PUC8, retrovirus. All three options are incorrect because these are
not suitable.
to deliver desirable genes into animal cells. So the correct answer to this question is option number 4. Let's move on to the next question.